Hi, everyone. Like Brady said, my name is Ben Stratton. I am a first-year grad student joining Dr. Richter's lab, focusing on the Copperhead system in Daniel Boone National Forest. So I realize now that Brady went over some of this stuff, but I am from Orlando, Florida. I grew up like 30 minutes away from where Dr. Watson grew up, which was pretty cool. Insert Florida man joke. I got my degree in environmental studies, concentrating in ecology and earth systems at Bates College in Lewiston, Maine. Like I said, I'll be joining Dr. Richter's lab. I am really big into ectothermic species. I think that temperature regulation that they do is a super interesting behavioral aspect. So I have come here specifically for copperheads, but all things ectothermic are really interesting to me. That is me working with one of our Burmese pythons at the Kentucky Reptile Zoo last summer. And this is one of my favorite diagrams about wildlife, of how to tell if a snake is dangerous. And a fun fact about myself is a really good friend from school is Sir David Attenborough's grandson. So if any of you guys are working on any documentaries, let me know. So Brady asked me to include a little bit about how I kind of got started into biology and field work. So I've always really been super interested in living things, specifically animals. I just think the fact that we are a bunch of cells made of a bunch of different chemicals that are now in some way thinking and talking to each other, I just think that's fascinating. So that is a big aspect of it. Additionally, I'm really interested in conservation biology. We are in the Anthropocene, and there every day, we're learning more and more about how we're actually impacting our planet and the organisms on it. And in order to actually do anything about that, we need to understand the organism's life history, their habitat requirements, and their behavior. It's also just really, really cool. So the first time I really got to do field work was through my abroad program. So I did the School for Field Studies program in Tanzania. So my work there focused on looking at how land use affects reptile species richness and biodiversity. So this is me right here. This is Dr. Ole Teisinger, who was my faculty advisor. And this is a photo of our field crew in Yaita Valley, which is in northern Tanzania. These are also some cool photos that I took. I think I look at these and it just kind of reminds me what it's like to be in the field. I think that was really the aspect of biology I fell in love with, going out, actually measuring something, coming back and discovering what does that mean. So for my undergrad work, I worked with Dr. Don Dearborn, who is the chair of the bio department at Bates College and an associate editor for ornithology. So a lot of his work focuses on host-parasite relationships in birds. And we came up with this study for my senior thesis. What this one was specifically looking at was the relationship that's been proposed between nest sanitation behavior and parasitic egg rejection. So just for a little bit of background, avian brood parasitism is basically just parasitism through laying your eggs in another nest. And this can be either interspecifically or conspecifically, so it can happen between species or within. So this is a cowbird that has parasitized the nest of an eastern phoebe. So you can see that's the cowbird egg. It sticks out pretty readily. But it can also happen conspecifically. So these are actually both American coots, and this is actually a nest that has been parasitized by the same species. So it can happen either way. Obviously, it's a lot harder to tell if you're being parasitized by the same species. And that was a big reason why herring gulls were our focal species. So doing this, it allows you to pass on the genetic information without having to deal with actually rearing your own kid, because that's a very energetic process. You've just created this baby, you've made the egg, you've incubated it, and now you have to raise it. Instead, once it's done, you push it off on somebody else. Which means that the accepting parents, if they do accept the parasitic egg, they incur those costs associated with raising that chick. And this exerts a selective pressure for these birds to be able to realize that they have been parasitized, and if possible, remove the egg. And evolutionarily, this has led to relationships this has led to parasitic egg rejection in some species. There's still a lot of variation, both interspecifically and conspecifically, of how individuals and species react to parasitism. Oftentimes, this variation is attributed to evolutionary lag. So the parasite has developed an evolutionary strategy that allows them to parasitize. The host species has not quite gotten there yet to be able to recognize it. But there are other factors that may affect this besides that evolutionary relationship, especially on the individual level as opposed to a species or population level. So a big thing of this would be learning or past experience. If they've encountered parasitism before, there's a chance that they recognize that if another bird is by their nest, this may be happening. Hormone levels were something that we wanted to look at, and unfortunately we were not able to, but there's been a lot of work that shows rejection is, there's differential amounts of rejection of parasitic eggs depending on hormone levels of the parent birds. And I know they've done this experimentally. I'm blanking on what hormone they were actually looking at, but the hormone level does have some effect. The morphology of the clutch is another thing. So if you have 3 eggs, in the case of a herring gull, which normally is 2 to 3 egg clutches, if all of your eggs look exactly the same, it might be easier to see one that sticks out. If all of your eggs are a little bit more varied, it's harder to see a parasitic egg. And then the final one, interactions with parasitic individuals, which is similar to the learning aspect. So when thinking about it in the framework of both evolution and experiences that could happen within an individual's lifetime, there's both an ultimate and proximate relationship between nest sanitation behavior and parasitic egg rejection. So what is nest sanitation behavior? Basically, this is the removal of debris from a nest. And this is things like fecal sacs, plant matter. In a lot of the herring gull nests we looked at, plastic was a big thing in there because they are nesting on the shorelines. And basically the entire idea of nest sanitation behavior is to increase reproductive efficiency, because these objects could damage eggs, they could facilitate microbial growth. There's a lot of different things they could do, interfering with how well you can incubate your eggs. If there's something blocking the brood patch from the actual egg, you're not going to get as good incubation and might not have as fit of offspring. Another big connection between nest sanitation and parasitic egg rejection is that they both require this ability to recognize what is your egg versus what is not your egg. And that's the first step. The second step is, once you recognize that, you need to be able to physically get whatever it is out of your nest, whether that be a parasitic egg or whether that be fecal sacs, eggshells, something else that is undesirable. So the nest sanitation hypothesis is an idea that it has both an ultimate and approximate aspect to it, but ultimately nest sanitation behavior might be the pre-adaptation for parasitic egg rejection, in that before you can recognize and remove something that looks like your egg, you need to be able to tell that something that doesn't look like your egg is there and remove it. In a more approximate sense, it has to do with a kind of mechanical link between actually performing the behavior, performance of sanitation behaviors and rejection. Sorry, I had a typo there. It predicts this link between the two behaviors in a more mechanical sense of an individual as opposed to evolutionarily. And this is through 2 possible avenues. The first one is recognition, where nest sanitation behavior may cause the host to actually pay more attention to what's in their nest and thereby become more likely to reject the parasitic egg. The other option is priming, where as opposed to the observing of something in the nest, it's the actual action of removing that may elicit an increased reaction to a parasitic egg. So as just a quick summary of that, the nest sanitation hypothesis, posits that nest sanitation behavior is this evolutionary prerequisite for parasite defense. And that species or individuals that reject parasitic eggs, if the nest sanitation hypothesis is true, the ones that reject parasitic eggs should have higher frequencies of nest sanitation behaviors. And in our experiment, what we were looking at, and which we'll get to more in a sec, is that sanitation may, if this hypothesis is true, actually prime the rejection of a parasitic egg, but there's a lot of really inconclusive evidence. Most of the work done with this has been looking at interspecific parasitism, specifically hosts that are victim to cowbirds or cuckoos. So we wanted to look at this in a conspecific species, and we wanted to basically see if our results would look similar based on what we got versus these other experiments that had very similar methodologies. So our study is we tested the nest sanitation hypothesis in the American herring gull, which in the United States is classified as a subspecies of the European herring gull. In Europe, it's recognized as its own species. So that was fun trying to decide which one to use. So it is believed to experience low levels of conspecific brood parasitism, particularly on their nesting sites. So they are colony nesters. So this was all on one island with hundreds and hundreds of nests on it. So our experiments were both conducted at the Bowdoin Scientific Station on Kent Island. So you can see the coast of Maine. This island right here is Grand Manan Island. We took a ferry to Grand Manan and then took a 2-hour ride on a lobster boat to Kent Island, which is a really, really tiny island, big herring gull nesting colony. And the only actual building on the island is the scientific station. So it was a really cool place to work up in the Bay of Fundy. So our experiments, there are 2 parts of them. The first one was looking at what physical factors of an object elicit removal from the nest, because we wanted to find something that we could use that we knew would be sanitized super frequently. So our question was, how do these factors affect rejection? And we did this by placing objects of various size, shape, and color into the nests, giving them 24 hours, and then recording how frequently each kind was rejected. So the objects were either red or tan. The color red we used for this and all other experiments is actually the same color of red that you see on the bottom of a herring gull's beak. And so there's a little red spot there that chicks will actually peck at the bottom of their parent's mouth, and that's how they know they want to be fed. So we used that same red color, and the tan color we used was the same base as a herring gull egg. So they are either red or tan. They were either egg-shaped or block-shaped, a little wooden cube, and they're either small or large. I believe for the large one, we used one that was herring gull egg size. The small ones, I believe we used a common eider-sized egg. And again, the purpose for this was we wanted to measure the baseline rejection of a non-mimetic gull eggs and identify one object that was frequently removed. So what we learned, and there's a more clear graph on the next slide, most of the things that they rejected were small. This is pretty consistent with the literature on how size affects this. Red and tan, there was less of a difference. Red ones were rejected more frequently, but not in a significant manner. And shape was very, very important. Wood, or we classified as wood because they were wood blocks, but the block objects were rejected far more frequently than any of the egg-shaped objects. So this is a combination of that data. So looking at this, we were able to decide that what we wanted to use for the sanitation part of it was the small red block, because we knew they could see it based on it being the same red color that's under their beaks. And based on this experiment, we knew that they are both able to recognize it and also physically remove it, because that's another component of it. And so using these results, we informed our decisions for the second part of the experiment to use the small red wood chip as our priming object. So our experiment 2 was actually testing whether or not this sanitation behavior primed parasitic egg rejection. So the nests were either randomly assigned to the control treatments or the control or experimental treatment. So nests in the experimental treatment, we simultaneously placed a red herring gull egg and a red wood chip. These eggs, we 3D printed them. They were the same shade of red, made to the same specifications of a herring gull egg. We actually made them lighter though, so we knew that they would be able to mechanically move them. And nests in the control group received only the egg. So again, we were testing for whether or not the addition of that wood chip primed the egg rejection behavior. So after we placed them, we waited a 24-hour period. Other studies used a longer period, but on Kent Island, it is very, very small. It's in the Bay of Fundy. There's a lot of tidal action and a lot of wind. And we were concerned that if we left them out longer than that, the possibility of them being blown away, or if there was a particularly high tide being swept up, we didn't want to deal with that. So we shortened the sampling period. And so here are two pictures of those nests. So in picture A, this is a nest where they sanitized the object, but did not remove the parasitic egg. And option B is they removed both of them, so they're ground nesters. You can see in the little nest right there, both of them have actually been taken out. We made sure to place them very closely to the center of the nest without actually damaging the current clutch. That way it would be fairly obvious if they were removed. And so we placed these, waited 24 hours, came back, collected, placed another set, waited 24 hours. And that other set was with new nests. We kept track of which nest we were using in order not to accidentally replicate. So our results showed that there was very little impact. The sanitation behavior had very little impact on the actual egg rejections. We had rejection came in 5 of each kind of nests. So it was 5 out of 40 nests for the treatment, and I believe 5 out of 49, 49 nests for the control. And within the experimental group, there was out of the experimental group, there were 8 that didn't actually sanitize at all, and 2 of those did remove the object, so we could not include those in this analysis. But this indicates to us that there didn't seem to be any priming effect of sanitation behavior. One of the cool things we did notice was that the presence of a gull at the nest at the time that we placed the objects did have a fairly measurable impact on whether or not rejection occurred. So even though the nest sanitation hypothesis doesn't appear to hold true in this species, there is some level of individual recognition that seems to be happening when we look at how they reacted to parasitism when they were either present or not present. So one of the things that we discovered is the egg rejection was not affected by either the visual stimulus of the egg or the actual performance of the sanitation behaviors, which is both the priming and the recognition concepts of the nest sanitation hypothesis. So we found sanitation occurring fairly widespread. I should have actually put the number, but sanitation was very, very widespread in the experimental group. However, it was not equally widespread with the actual removal of parasitic eggs. So because of that difference, we assume that it may not be associated. These behaviors may not be associated. One reason that we think this is possibility of rejection error. So eggs are obviously very costly to make in terms of reproductive energy. And if you have your own eggs and you think there's a parasitic one, there is a chance that you will reject one of your own eggs, specifically in a conspecific setting, when your eggs look more similar, even though our eggs were bright red. There is definitely something that they're worried about, and we attribute this to rejection or worry of rejection error. Because of what I mentioned earlier, that the eggs were light and easy to move, and we used the red color that we know that they were able to see, we were able to kind of cross out inability to move or inability to detect as reasons. And it seems more likely that this failure to reject eggs is kind of part of this decision rule. And this agrees with a lot of the current literature that being egg shaped is one of the more important factors when it comes to parasitic egg rejection. So it may just be that this species, they can tell something's wrong, but they would rather accept that cost of a potentially parasitic egg than try to reject it, and in the process either damage one of their own or even possibly reject their own egg. Additionally, there are some cases of herring gulls being parasitized by common eiders. But we see in precocial species like this, meaning the herring gulls are fairly active as young. They don't need a tremendous amount of help the way some other species do. It's more common to see facultative brood parasitism. So where cowbirds and cuckoos are obligate brood parasites, that's their primary method of reproduction. They dump their eggs in another nest. In a lot of these species that are more precocial, you see it happen more facultatively or opportunistically, where a parent is trying to get rid of an egg on an opportunity basis as opposed to their main strategy. And compared to parasitism that happens interspecifically, again, most of the work has been on cuckoos and cowbirds. It's a lot less expensive for the parent energetically to accept that baby. I need to brush up on which one actually does it, but either cuckoos or cowbirds actually push the other eggs out of the nest. Whereas a herring gull, if this happened, you would probably just have one more mouth to feed. So there is kind of a difference in how high the stakes are for actually being able to recognize that parasitic egg. So like we pointed out, non-mimetic egg rejection is 4 times higher at nests when a gull was present. So it is really hinting that this witnessing a disturbance might be an important factor in this decision rule to reject. And especially in a colonial nesting setting where the herring gulls were, where there are hundreds of nests on the ground, that interaction of actually seeing it, because all your eggs look the same, might be the most important thing, or it just might be a very important factor. So our research leaves with kind of several different options where we could go. So one of them is looking at how nest density impacts likelihood of rejection. So there was done, I believe in the Czech Republic, work done on black-headed gulls that found that both likelihood of conspecific parasitism increased and likelihood of rejecting parasitic eggs increased as nest density increased. So as there are more nests per area, there is more likely both to be parasitism, and birds there are also more likely to reject parasitic eggs. Also, potentially environmental factors like temperature of the eggs or other things like that we didn't have the chance to look at within our study period. We could analyze variation between sexes because there is some difference in nesting behaviors and how they incubate and how they kind of their range during this breeding season. So there may be some differential things that we were not able to look at because we didn't sex the birds. And I also think it would be a really good idea to try this with eggs that maybe looked a lot more like herring gulls and see if you see a similar pattern. That was it. Does anyone have questions? Sorry, that ended kind of abruptly. And this is that's actually me collecting data there wearing lobster pants. This is the island when the tide is high. That's like 30 feet of water. We could actually walk from here to there during low tide, which is pretty cool.
Cool stuff, Ben.
Thank you.
Mr. Knight, yeah, we're going to open it up to questions now. So first we'll do some in-person questions. So for folks that are here, you can raise your hand and ask a question. And then, so while we're doing that, folks who are watching via Zoom, if you could get some questions ready, if you have, and put them in the chat. We're going to take those after the in-person questions. So when somebody here asks a question, Ben, I'd ask you, just so we can hear the question loud and clear for the people who are going to be on Zoom or going to be watching the recording, could you please repeat the person's question before you answer the question?
Absolutely. Alex.
So I know I promised no hard questions. This is genuinely interesting. So when you were trying to determine whether they used sanitation behavior, why didn't you do that prior to assigning treatment? So put a block in a bunch of different nests, see if they do sanitation behavior, and then use that as a baseline for placing the eggs in, say, a sanitizing group and a non-sanitizing group, as opposed to putting a block and an egg in every nest. Does that make sense?
So we did have a group with just an egg and then a group with an egg and the block. Sorry, I'm a little confused by your question.
I guess why not determine prior to your experiment whether an individual, what the frequency, or whether an individual bird is a cleaner or not a cleaner, essentially. And so see, you have these nests with an egg and a block in them, right?
Mm.
And presumably, if they don't get rid of the block, they're not a cleaner, right?
Well, the thing is, we at this point didn't know what their reaction was going to be at all. No one has looked at nest sanitation or parasitic egg rejection in American herring gulls. So we didn't know what their baseline reaction to any of this would be, which is why in experiment 1, we tried to cover as many nests as possible. I think we used 160 nests. And using all those different object types, that's how we collected data on something that was sanitized frequently, because most birds have some kind of sanitation behavior to help their reproductive efficiency. So we basically looked at the literature, found what factors of an object tended to elicit a higher rejection, because the entire purpose of that first one was we wanted something we knew they would reject or they would remove very readily. And so the small red wooden block was rejected in I believe it was in 90% of the nests that we used. And then after that, that was when we actually separated out the nests, gave them a time period between experiment 1 and experiment 2, and then assigned them as either just the egg or egg and the wood. Does that answer your question?
To an extent, yes.
Okay, sorry.
No, you're good.
So yes, so she asked if this study is ongoing or being replicated. Right now, I don't know. I know Dr. Dearborn is on sabbatical right now, and he is trying to get funding to go back and do this. But we actually were operating out of, so Bowdoin College actually owns Kent Island, even though it's in Canada. So we were working through them, and based off those results, we may or may not continue it, just based on kind of how useful the results end up being, because there's a lot of different research. There's some stuff with savanna sparrows and some stuff with common eiders that they're also doing here. So I think that if the study were to be continued, it would be, great to go back here and look at this population as well as other ones. But as of now, I don't know, just because COVID has kind of thrown everything off. Yes.
Good job, by the way. That was cool.
Thank you.
Stuff and super amazing place, and they find it's really neat. I remember as a kid almost getting swept away by the big high tides that come.
Yeah.
So I was just going to ask about like nest failure or nest abandonment, that putting objects, and in some species, when you whether you do it as an experimentalist or if some of our birds are parasitizing nests, that it can be the females or the hosts to abandon the nest. Did you see any evidence of that? Or and kind of related to that, it seems like putting a red object in the nest might potentially attract predators, nest predators, and kind of how did you observe that, or how do you know?
So regarding that, regarding the abandonment question, we didn't actually see any instances of nest abandonment, which was pretty interesting, because on our data sheets, we compiled a whole category just specifically so we could keep track of that. We didn't have any instances of nest abandonment happening. And I don't know exactly why they decided not to. One of the things that we did do is we specifically chose nests that had a full clutch. So possibly they were less likely to abandon because there was a full 3-egg clutch in there. Regarding predators, I don't actually know if they have predators on Kent Island at the colony. I mean, I'm sure there are some, but it's the only mammals that I know are on the island was I believe a species of rabbit that someone introduced and had kind of proliferated. But we didn't see any evidence of predator activity in the area. So yeah, we didn't get any of that. Yes.
All right, does anybody else have any in-person questions? So move on to the chat questions. So one of our questions comes from Dr. Oliver Oakley here, and he asks, did you examine multiple sanitation events for specific nests? And if so, would it have been better to use those nests to test for priming for removal of parasitic eggs?
So in relation to multiple sanitation events, sorry, I'm a little unclear on what that means, but they all only had one piece of the red bark or the red wood chip. So we actually did not stay and monitor the nests. So the only way we could really tell was once we got back, because there has been some evidence suggesting that behavior might be affected if researchers are fairly close. So we tried to go out early in the morning, place everything, give them a full 24-hour window, and then come back, and whether or not sanitation or rejection occurred was based off of placement of the egg after we got back. And again, there has been different time frames used in this experimental design. I know with barn swallows, I believe they used two or 3 days in a very similar study. But again, with the conditions on Kent Island, we wanted to try to give them long enough to recognize and reject it, but also keep it constrained so that environmental factors like tide and wind wouldn't possibly lead us astray when looking at whether or not sanitation or rejection occurred.
So to clarify the multiple sanitation events, Dr. Oakley wrote, to repeat, single in same nest. So single sanitation behavior in the same nest.
Sorry, I'm still a little.
I think you pretty much answered the.
Okay, yeah, there only was one sanitation behavior per nest because we tested each nest once,
Okay.
kept track of it via GPS, and then went to the other nests kind of spread around the island, each group randomly using nests that were selected on different parts of the island to try to get an overall view. But each nest was only tested once. So if an individual did sanitize, just due to the kind of time frame that we had to be on the island, we weren't able to go back and look among individuals that sanitized, was this a higher proportion of rejection.
And we have another question from Dr. Mott, and he asks, I might have missed this if you mentioned it, but is there a stable rate of brood parasitism in these populations, i.e. percent of nests that are parasitized in a given season, year, etc.?
So for herring gulls, no, there isn't any number that anyone has. There was that study I mentioned that was done in the Czech Republic looking at black-headed gulls, which are in the same genus. I believe, I don't have that paper up right now. I believe that was in the 5 to 10% natural parasitism rate. And that was the one that rate of parasitism increased as density of nests increased. But with the herring gulls, we were actually the first one to look at this behavior in them.
Any further questions from folks in here or on the chat? People who asked questions on the chat gave thumbs up, so it's like,
All right,
they liked your answer.
I'll take it.
All righty. And with that, I think we'll go ahead and close for today. Thank you.